Hello, my name is Taylor Petrie, and I'm an 11th grade student at Franklin High School. I am part of the Biomedical Science program, also known as Project Lead the Way. I was inspired to enroll in this program by my older brother's prior participation. I was skeptical at first, as I've always wanted to be a veterinarian, and I didn't think it was necessary to study the human body, but over time I've grown to love this program and use it as a foundation for my career aspirations. PLTW is a 4-year program, and currently in my junior year, our focus is on medical interventions. Our introductory lesson revolved around investigating a college outbreak. Each of the 7 individuals we were assigned had unique symptoms, risk factors, and relationships with one another. Our task was to analyze the data given to find out who spread the disease. In order to appropriately assist our patients, we had to find an answer for the disease that was causing the problem after gathering our hypothesis. We studied how DNA is sequenced using the polymerase chain reaction, a laboratory technique that amplifies particular DNA segments into millions or billions of copies. We also learned how sequenced DNA can assist us when identifying medical problems. When you plug a DNA sequence into a database, such as nucleotide blast, multiple results in comparison to your sequence show up, and certain factors, such as the e-value, allow you to identify what is your likely cause. Neisseria meningitis, a bacteria that lives in the back of the nose and throat, was found in one of our patients when we used this kind of database. Finding the source of the other patient's illnesses was our next task. We used enzyme-linked immunosorbent assay, also referred to as ELISA. This method uses an enzyme to provide a measurable signal, in this example, for us, a color shift, in order to identify and quantify molecules such as proteins, antibodies, and hormones. This test allowed us to determine whether other patients had the same disease. Next, we focused on the different parts of a bacterial cell and whether they are gram-positive or negative, and how different antibiotics work when fighting against these cells. We conducted another experiment while keeping this knowledge in mind. We possessed 2 strains of E. coli. Strain 1, which is resistant to streptomycin, and strain 2, which is resistant to ampicillin, 2 types of antibiotics. On Petri dishes, we streaked the strains together to see if one strain could transfer its resistance gene to the other, creating a new strain resistant to both ampicillin and streptomycin. Antibacterial resistance was demonstrated through this experiment. We are now diving into the components of the ear and how they work. The things we learned throughout this program are beneficial to our everyday lives, as we have developed advanced understandings of our own bodies. Because of both my passionate interests in science and the hands-on learning we perform, I've been able to thoroughly understand the concepts each year, and even if students don't want to work in the medical field, this program offers a lot of opportunities. Thank you so much.